All right, good morning, everybody. Are we ready to go? Okay, so I just wanted to remind you again the cholesterol synthesis that we finished last time. Um, the cholesterol, remember, in our body, right, half of the cholesterol in our body is synthesized de novo. Our body is able to synthesize half of cholesterol, and the other half of cholesterol is actually comes from the diet. And in terms of synthesis of cholesterol, the starting molecules or the precursors of cholesterol is acetyl-CoA, right? Remember, so acetyl-CoA is very important molecule, can be utilized for synthesis of cholesterol and what other molecules as lipids that can be synthesized from acetyl-CoA. We all talk about it already, right? Fatty acids, right? Fatty acids, cholesterol, and what else? The very important fuel, energy fuel during the fasting, we utilize ketone bodies, yes. So remember, acetyl-CoA is a key molecules in our body. It's a metabolized, right? The intermediates of the glucose, amino acids, and fatty acids. All these three nutrients can be oxidized or metabolized to generate acetyl-CoA, right? And acetyl-CoA is important molecule that connect all three nutrient metabolism together. So acetyl-CoA can be used to synthesize cholesterol, fatty acids, and ketone bodies, right? And we also talk about the whole pathways of cholesterol synthesis involve 17 enzymes, right? And of course your NADPH and ATPs. But you just need to remember the key enzyme for cholesterol synthesis. Which enzyme is the key enzyme for cholesterol synthesis? HMG CoA reductase, right? Yeah, so this is a key enzyme for cholesterol synthesis. And also try to remember what factors includes hormones and metabolites, intermediates that regulate HMG-CoA reductase. How insulin regulates HMG-CoA reductase? Increase, right? And what about glucagon? Inhibits, okay, yeah. And what about the key substrate that can regulate the cholesterol synthesis or HMG-CoA reductase? Remember this is one slide, right? Cholesterol. What's that? Thyroid hormone? Yeah, that hormone stimulates, right? And cholesterol inhibits and mevromet, right? It also inhibits the HMG-CoA reductase. And the drug that has a cholesterol lowering effect, right? Statins, the targets of statin is HMG-CoA reductase, right? Okay, so that's, I think, is most important information that you guys need to know for cholesterol synthesis, right? And we also start to talk about the lipoproteins, right? So the lipoproteins have the different type of lipoprotein that in our body. And the main purpose or main function of lipoproteins is, what are actually the main functions of lipoproteins? Why we need lipoproteins? To help transport lipids, right, between tissues. So, and where actually lipoproteins are located? Primarily, in which part of the body? Plasma, circulation, right? So lipoprotein majority of, the lipoproteins are present in the plasma, in the circulation, and the purpose of that we have a lipoprotein is to help transport lipids between tissues, right? Of course, in the circulation, right? To transport lipids from one tissue to the other tissues. And we also talk about the different type of lipoproteins and structure of lipoprotein, right? The lipoprotein basically has a core, lipid core, and the surface. So surface lipid core can have, depend on which type of lipoprotein. So core lipids can have a triglyceride or cholesterol esters, and core, the surface basically is the phospholipids, monolear phospholipids, also has a apoprotein, right? So apoprotein so it's sort of kind of structure of the lipoproteins. And different lipoprotein has a different type of apoprotein on its surface, right? And we also mentioned that the function of apoprotein on the surface of lipoproteins mainly is to help the to keep the structure integrity, right, and to stabilize lipoproteins, and also to help the suspension of lipids in the plasma. And most importantly, the two important functions that we mentioned, that apoprotein is also served as important cofactors for enzymes, right? And one apoprotein that is very important for lipoprotein lipase activity is ApoC2, right? ApoC2 is a apoprotein that present on the surface of certain lipoproteins that is very important for lipoprotein lipase LPL activities. So if someone does not have enough ApoC protein or some ApoC protein deficiency, then we're causing some problems that we're going to talk about later, right? So the LPL cannot function well if we don't have enough ApoC on the surface of lipoproteins. And also, the other lipoproteins like ApoE or ApoB100 are the ligands for the LDL receptors, right? So LDL receptor is a receptor that on the surface of peripheral cells that help recognize lipoproteins LDL and pick up LDL, right? So LDL receptor is critical for uptake of LDL into the cells. So ApoE is functioning as the ligand that can recognize the help receptor to recognize, that can be recognized by receptors, right? So if the lipoproteins, again, does not have a ApoE on a surface, then this lipoproteins cannot be recognized by LDL receptors, so it cannot be picked up by the cells, right? And composition of lipoproteins, right? We also mentioned that the two apoproteins we called triglyceride-rich apoprotein, like chylomicron and VLDL. The reason is because these two type of lipoproteins, and the majority of lipids in the core are the triglyceride. So in other words, these two type of lipoprotein, their main function is to help transport the triglyceride, right? And LDL and HDL are the cholesterol-rich lipoproteins, and it also means that these two type of lipoproteins helps transport the cholesterol. And we also mentioned that the apoprotein, like different type of apoprotein present in different type of lipoproteins. For example, chylomicron has actually 1, 2, 3, 4 different type of apoprotein on its surface, and ApoB48 is specific to the chylomicron because ApoB48 is synthesized in the small intestine, right? So it's together with the chylomicron. Chylomicron is also mainly synthesized in the small intestine. So and VLDL, the main type of ApoB protein is ApoB100. So ApoB100 is synthesized in the liver, and similarly, the VLDL also synthesized in the liver. So this, you can tie this together, right? So again, different type of lipoproteins contain a different type of apoproteins. And you also probably already noticed that the ApoC2, which I just talked about, right? It's very important, the cofactors for LPL. And what is the LPL function? Do you guys still remember? What is the function of LPL? Yes.
It hydrolyzes the free fatty acid and
Triglyceride, right? Triglyceride. Yes, yes, yes. So where is the triglyceride, located? That can be hydrolyzed by LPL.
Yeah.
Triglyceride can be in the, yes.
Is it in the liver?
Not quite.
Adipose?
But LPL, triglyceride can be present in the cells, right? Like in fat cells, and also can be present in the lipoprotein, right? So remember we just talked about, I guess, a couple times right before, the lipoprotein lipase LPL basically hydrolyze triglyceride in the lipoprotein, VLDL and chylomicron, right? Does that make sense? So and we also just talked that ApoC2 is required for LPL function. So do you think that makes sense? So ApoC2 localized in both chylomicron and VLDL, right? Because LPL basically hydrolyzes triglyceride in VLDL and chylomicron. So that also need ApoC2 on the surface of these two type of lipoprotein. So then ApoC2 and LPL can work together to hydrolyze triglyceride in these two specific type of lipoprotein, right? Does that make sense? So probably, I hope this will also help you to memorize, this ApoC2 function in helping LPL activity, right? And help LDL hydrolysis of triglyceride in VLDL and chylomicron. Okay, remnant of lipoproteins, and we also talk about it, that the chylomicron basically synthesized in which tissues? Small intestine, right? And where is triglyceride that inside of a chylomicron coming from? Diet, right? So the dietary lipids, triglyceride basically, right? Once it's absorbed into the enterocytes, right, which is a epithelial cell in the small intestine, and then re-acidified back to triglyceride, and this triglyceride need to be transported to its destination, basically the adipose tissue for storage, right, in the fat condition. So chylomicron is a primary lipoprotein that is produced or made in the small intestine in enterocytes, then help triglyceride, dietary triglyceride transport. So chylomicron is also synthesized in enterocytes and pack the triglyceride, right, into the chylomicron, and then leave the small intestine and into the circulation, and then transported the dietary triglyceride to the adipose tissue, right? And the adipose tissue pick up most of the triglyceride in the chylomicron, but still have some triglyceride left in the chylomicron, right? So so this might, once the triglyceride pick up by the adipose tissue, and the leftover of triglyceride in the chylomicron, and this called chylomicron remnant. So chylomicron remnant basically is smaller in terms of the size, right, and also have a less triglyceride in the core, and eventually the chylomicron remnants are picked up by the liver, and liver can take up the leftover the triglyceride in the chylomicron remnants. And you also know, right, this LPL, lipoprotein lipase, and it's critical to break down the triglyceride in the chylomicron into fatty acids and glycerol, and fatty acids then be able to be taken up by the fat cell, the adipose tissue. So what will happen if LPL activity somehow is defective, as lower or something like this? And how about the chylomicron levels in the circulation if LPL cannot function well? High, right? So because you can't break down the triglyceride efficiently, so triglyceride cannot be taken up by the fat tissue. So you have a higher chylomicron lipoprotein levels, and you also have a higher triglyceride levels in the circulation, right? So LPL, again, is very critical, the enzyme that helps adipose tissue to take up the triglyceride, fatty acids. Okay, now chylomicron. So core lipids. What are the core lipids in the chylomicron? Core lipids in chylomicrons. What are the core lipids? Actually, two types of core lipids, right? Two types of lipids in the core, the lipoprotein: triglyceride and then cholesterol ester. can you guys share me what type of lipids in the Carmichael core? Which type of this is the majority lipids in the Carmichael? Triglyceride or cholesterol esters? Well, Carmichael? Triglyceride. Yes. I mentioned that Carmichael and VLDL are the triglyceride-rich lipoprotein. That means in the core, these two type of lipoprotein have more triglyceride, right, and less cholesterol compared to the other two, compared to LDL and HDL. Does that make sense? Right? So the core lipids in Carmichael are triglyceride and cholesterol ester, but I just wanted to remind you that triglyceride should be the major type of lipids in the Carmichael, right? And apoproteins, if you go back to that table that that we showed before, what type of apoproteins that are on the surface of Carmichael? What type of ApoB? 48, yes, ApoB 48, right? Apo C2, of course, right? It's and then Apo A1, 2, 4, and 3, and Apo E. Almost every type of apoprotein, right? So I just wanted you guys to remember ApoB 48, Apo C2, and Apo E, right? And this three type of apoproteins are very important for its function and synthesis. So where are Carmichael synthesized? In which tissue? Small intestine, right? Small intestine and enterocytes specifically are the major site where Carmichael synthesized, right? And where does T triglyceride in Carmichael come from? Diet, right? From the diet. And ApoB 48 is made from enterocytes, right? So again, Carmichael is a triglyceride-rich lipoprotein. The main function of Carmichael is to transport a dietary triglyceride, right, from small intestine to mainly to fat tissue and also liver. So Carmichael is synthesized in the small intestine and transport the dietary triglyceride. And ApoB 48 is the main ApoB protein, right, that is synthesized in the small intestine and is packed on the surface of Carmichael. And Carmichael also has a ApoC2, which is important for LPL function or activity. Okay, so now let's talk about metabolic fate of Carmichael or Carmichael metabolism. This is a pretty comprehensive diagram, so I hope that will help you to understand better the whole picture and how Carmichael is functioning and help help transport the triglyceride from the diet to the adipose tissue and the liver. So and start from the small intestine. When we eat, right, under the fat condition, and we absorb a lots of lipids, particularly triglyceride from the diet. And first of all, chylomicron is synthesized in the small intestine as so-called nascent chylomicron. That means some of the apoproteins are not present yet, in the nascent chylomicron. And once it's released into the circulation, and chylomicron then receive the other type of the apoproteins, for example, ApoC, ApoE, from HDL, right? And then become the mature chylomicron. Because of the, again, ApoC2 is important, right, for LPL hydrolysis of the triglyceride. So once it has a ApoC2, then it will help lipoprotein lipase, LPL, to hydrolyze triglyceride and release fatty acids and glycerol. And fatty acids is molecules are small enough so that can be taken up by peripheral tissue here, particularly the adipose tissue, right? So adipose tissue can take up fatty acids and then can re-acidify to triglyceride and stored in the adipose tissue, in the fat tissue, right? And still some triglyceride is left in the chylomicron. If you compare the size, between chylomicron remnant to chylomicron, it's much smaller, right? Because a lot of almost like 90% of triglyceride is already gone, right? It's already picked up by the adipose tissue. So the chylomicron remnant has less triglyceride in the core, but also lose the apoprotein A and apoprotein C, and this apoprotein will be giving back to the HDL, right? So, and finally, chylomicron remnant can be taken up by the liver, right? And liver has a LDL receptors, right? And LDL receptors can, have so-called the ApoB100, ApoE binding site. So this binding site can recognize the ApoE, right? Because of chylomicron remnant has the ApoE on its surface. So this ApoE protein can bind LDL receptor, right? ApoE binding site. So then it can be recognized and taken up by the liver, right? Does that make sense? Okay. So this basically is the pathways, how chylomicron is metabolized. Yes.
So what is the difference between the chylomicron and the chylomicron remnant? Is it the receptor for the
You mean the difference? Yeah, one difference is the lipids content, right? It have a less triglyceride, and then also the the apoprotein on the surface, right? They have a less apoproteins. So like the chylomicron, mature chylomicron has ApoB48, ApoE, ApoA, and C, C2 particularly, right? And chylomicron, only has B48 and ApoE. And the rest of the apoprotein, like A and C, will last, and basically is giving back to HDL, right? Any questions about the chylomicron metabolism? So you can see from the whole process, the triglyceride from the small intestine initially from the diet being transported to adipose tissue and the liver, right? So this is the main function of a chylomicron. So why we need to synthesize chylomicron in the small intestine? Again, the purpose of chylomicron is to transport dietary triglyceride from small intestine to adipose tissue for storage and also to the liver. And this just gives you sort of simple version of the chylomicron metabolism. And this is sort of mature chylomicron, right? It has everything, core lipids, triglyceride, and cholesterol esters, and then the apoproteins. And once chylomicron is transported to adipose tissue, and triglyceride in the chylomicron hydrolyzed by LPL. So 90% of triglyceride is taken up by adipose tissue, and also chylomicron loses the ApoA and ApoC, and eventually become the chylomicron remnant, and chylomicron remnant eventually be able taken up by the liver, right? And the liver actually has a different lipase that hydrolyzes triglyceride once it's picked up from the chylomicron remnant, which is hepatic lipase, right? So it's a little bit different here. The mature chylomicron LPL is the enzyme that hydrolyzes triglyceride, and in chylomicron remnant is once it's picked up by the liver, the triglyceride in the chylomicron remnant actually hydrolyzed by liver lipase, hepatic lipase, right? So which enzymes hydrolyze triglyceride in chylomicron? LPL, yes. And which enzyme hydrolyzes triglyceride in chylomicron remnant? Hepatic lipase, right? Hepatic lipase. Okay. Any questions? All right. Okay, now let's talk about the second type of a lipoprotein, VLDL, very low density lipoprotein, right? So the core lipids in VLDL, what are the core lipids in the VLDL? Triglyceride, right, again, and some majority of the core lipids are triglyceride in the VLDL, and also some cholesterol esters. And VLDL is also a triglyceride-rich lipoprotein. That means the function of VLDL is also to transport triglyceride. But the route, what I try to say is different. The VLDL, where actually are the VLDL synthesized? In which tissue? Liver, right? Okay, so that makes sense. So liver, VLDL actually transports triglyceride from the liver to the other tissues. What about the apoproteins on the VLDL? What type of apoprotein? Let's see. ApoB100, right? And ApoC2, which is also important for LPL activity, right? For LPL to hydrolyze triglyceride in VLDL, and also ApoE. And VLDL is synthesized in the liver. And where do triglyceride in VLDL come from? Right? VLDL basically transports triglyceride, right? And we know in the liver, right? Triglyceride synthesized in the liver, but it's a different, the fatty acids can come from the different sources, right? So let's see. So first, the de novo synthesis from dietary carbohydrate. For example, in the fat condition, when we have a lot of carbohydrate, right, absorbed into the body, we have a increased lipogenesis, remember, right, to convert carbohydrate to fatty acids. So fatty acids can be synthesized, lots of fatty acids can be synthesized from the carbohydrate, from the glucose in the liver in the fat condition. So fat condition will have lots of fatty acids available through the lipogenesis pathway. So, and these fatty acids need to be packed, need to be acidified triglyceride and packed into VLDL. And where do you think this, de novo lipogenesis, this lipids or triglyceride should go in the fat condition? Adipose tissue, right, for storage. So adipose tissue not only just take up the dietary triglyceride, but also take the triglyceride that's synthesized in the liver in the fat condition, through the lipogenesis process, right? So, so the de novo synthesized fatty acids or triglyceride in the liver need to be transported to the adipose tissue for storage in the fat condition. So this is one of the important sources of triglyceride that packed in the VLDL. And secondly, long-chain fatty acids internalized by the liver from the plasma lipoprotein remnants. For example, we just talked about chylomicron, right? In the fat condition, chylomicron remnants can also be taken by the liver. So liver also takes up some triglyceride from the chylomicron, and the chylomicron triglycerides are supposed to, go to the adipose tissue, but some accidentally, you have some kind of leakage, right? Adipose tissue can pick up at the first place. So this leftover triglyceride in the chylomicron takes up by the liver, and liver then repacked the fatty acids to triglyceride, and then to the VLDL, and then sent back to the fat tissue for storage, right? So this is sort of the second, sources of triglyceride in the VLDL. And the third sources of triglyceride is the unsterified or free fatty acids, that something happening in the fasting condition, right? When we have lots of fatty acids that being released from fat tissue, and liver picks up these free fatty acids from the circulation and reacidify to triglyceride, and can also be packed into the VLDL. And eventually, VLDL takes this triglyceride to, what do you guys think, to which tissues under the fasting condition? Definitely not the fat tissue, right? Fasting condition, we just mobilize the fatty acids, right? I mean, triglyceride from fat tissue to mostly to the muscle, right? Because muscle need to utilize fatty acids. So this third sources of fatty acid basically from the triglyceride breakdown, triglyceride mobilization from the fat tissue, right, during the fasting condition, and packed into the VLDL, and VLDL takes this triglyceride to the muscle. And muscle also has a lipoprotein lipase, LPL. So under the fasting condition, the muscle LPL actually activity is much higher. So then LPL can break down triglyceride in the VLDL and takes up the fatty acids for utilized as energy in the muscle, right? Does that make sense? I mean, I know it's very complicated, but is that clear? Right? So, and we're also going to talk about LPL, in adipose tissue and versus muscle. Its activity is regulation, in terms of regulation, activity is quite different. LPL in the adipose tissue basically is increased in the fat condition, can stimulate by insulin, which makes sense, right? The fat condition, the LPL in the adipose tissue need to be up, regulated, so then can break down chylomicron triglyceride, VLDL triglyceride, and to the fatty acids and pick up fatty acids for storage. Yeah.
Just as a reminder, is the LPL in the muscle and the adipose, is it secreted by the cells and then it actually does its action outside the cell?
Yeah.
Or is it so that the parts can come into the cell, so that the fatty acids can go right into?
Yeah, that's a great question. So LPL actually is secreted, outside cells, actually localized in the, the artery, well not artery, like a capillary wall, epithelial cell. So then actually facing the circulation, right? So they're able to contact the, chylomicron or VLDL, so able to hydrolyze triglyceride. Yeah, that's a good question. So again, I mean the muscle LPL and fat tissue LPL, they are regulated differently. They're actually opposite. For example, fat condition, fat tissue LPL is upregulated, and during the fasting condition, the muscle LPL is upregulated, and the functioning also is different, right? Okay, yeah.
Could you repeat what happens
In the fasting condition, yeah. Okay, sure. So during the fasting condition, right, because we know that triglyceride need to be break down from the fat tissue, right, and release the fatty acids into the circulation, because a lot of other tissues need to utilize fatty acids as an alternative fuel, right? We don't have enough glucose, or we want to spare glucose. So during the fasting condition, the fatty acids level in the circulation increase because of lipolysis increase, right? So you release fatty acids. And these fatty acids can be taken up by the liver, and the liver reacidify fatty acids to triglyceride, and this triglyceride packed into VLDL, and VLDL take this triglyceride from the liver to the muscle, right? And it's still in the VLDL. So, and once this VLDL arrive in the muscle, then muscle LPL can hydrolyze triglyceride in the VLDL, right, into the fatty acids, and then fatty acids can be eventually taken up by the muscle for energy production, for ATP production. So this is during fasting condition. So VLDL can be high in both fat and fasting condition, but the purpose is different, right? So in the fasting condition, VLDL basically be used up as energy, and in the fat condition, triglyceride and VLDL basically is taken up by the fat and stored, in the fat tissue. All right, does that make sense and is that clear? Okay, let's see. Okay, that I guess it was time for a break, right? Okay, guys. Okay, now let's move on. All right, so VLDL metabolism. How VLDL is metabolized? Again, VLDL is the role of VLDL is, or the purpose of VLDL is to transport triglyceride mainly, right, from liver to the other tissues. In the fat condition, can be to the adipose tissue for storage of triglyceride, and during the fasting, it can be transported to the skeletal muscle for utilization of the triglyceride fatty acids as energy. So, and in this slide, I believe that primarily talk about the, didn't talk about the fat and fasting condition, but just I guess the transport between liver and adipose or skeletal muscle, like heart muscle. So the VLDL basically is synthesized in the liver, right? Pack the triglyceride, and triglyceride can have three different sources, right? It comes from three different sources. One is from the carmichael remnant, initially from the diet, and the second is from the lipogenesis, de novo lipogenesis, right? And the conversion of dietary carbohydrate to the fat. And lastly, the fatty acids can be also from the triglyceride breakdown in the fat tissue during the fasting condition, right? And so eventually VLDL is synthesized in the liver under different physiologic condition. I mean, fat fasting condition, VLDL can all are able to be synthesized, right, and pack the triglyceride. And again, at the first time, at the initial stage, that the VLDL is synthesized as the similar to the chylomicron in the form of the so-called nascent VLDL, and also because of this nascent VLDL lacks the ApoC and ApoE, right? So, and once it's received more ApoC and ApoE from HDL, and VLDL become mature, right? So eventually mature VLDL, once it's arrived in the peripheral tissue or like in the adipose tissue or muscle, then the triglyceride can be break down by LPL, right? And again, ApoC2, which on the surface of VLDL is important for the LPL function. So LPL again can be synthesized from both adipose tissue and the skeletal muscle, depend on again the physiological condition. But anyway, the function is the same, right? LPL always be, able to hydrolyze triglyceride, release fatty acids and glycerol. So fatty acids can be taken up by the adipose tissue in the fat condition, can be reacidified to triglyceride and stored in the adipose tissue. And if it happens in the skeletal muscle, the fatty acids being oxidized and for ATP production under the fasting condition, right? And VLDL eventually become a VLDL remnant, which is IDL. So you can see IDL or remnant, VLDL remnant, is also have a smaller size because of lots of triglycerides being also taken up by the tissues, right? So and also giving the ApoC, ApoE back to the HDL. So eventually, the IDL can also be further changing the lipid content, right, in the core and eventually become the LDL. So there are also so-called lipid exchange happening in the circulation between the different type of lipoproteins, particularly between HDL, VLDL, IDL, and LDL. Right? So lipid changing means that some of the lipids, for example, triglyceride, can be transported or transferred from IDL or LDL, HDL to HDL, and the cholesterol esters that can be transferred from HDL to IDL, LDL. So LDL eventually become cholesterol-rich lipoprotein. So you can see that almost have LDL almost has no triglyceride, right? Because triglyceride either be picked up by the tissues, adipose tissue or muscle, or being transferred to HDL, right? So LDL eventually become cholesterol-rich lipoproteins and only has ApoB100 apoprotein on the surface. So LDL is able to be picked up by liver and also other peripheral tissues, for example, arterial wall, epithelial cells, or macrophages. All right. Is that clear? Any questions about this metabolic pathways of VLDL? Any questions? Paul?
I wanted to ask,
Yeah.
I also see is the VLDL is broken down to fatty acids and to the IDL and HDL that are later turned into LDL. And also, do we take also into consideration glycerol?
Sorry,
Do we also
last
take
question.
into consideration glycerol coming out of VLDL or no?
You mean lipid exchange,
For the VLDL, when it's broken down,
right?
do we also take into consideration glycerol that it also breaks part of it breaks down into glycerol?
Visceral? I mean, I'm sorry.
There's the glycerol right there.
Want to go through again? I'm sorry. Visceral. What type?
Glycerol.
Oh, glycerol. Oh, okay. Yeah. So right. I mean, the LDL basically eventually, lose all the triglyceride. Right. Initially it was VLDL, right? So VLDL contains both triglyceride and the cholesterol esters. I know what's your question. You mean where's the glycerol go, right? Sorry. So glycerol basically is actually taken up by the liver, right? Under the fasting condition, right? So can be used for gluconeogenesis. And under the fat condition, actually glycerol can also be recycled, by the adipose tissue and used for synthesize of triglyceride, right? Yeah, sorry. All right, is that clear, everybody? Okay. Yeah, this again is the process, simple process in VLDL metabolism, right? The VLDL, mature VLDL, contains triglyceride, triglyceride esters, and the ApoB100, ApoE, ApoC2. And triglyceride in the VLDL can be break down by LPL, right, in both fat tissue or muscle, then triglyceride can be eventually picked up by these tissues. And also ApoC2 leaves the VLDL, right? So eventually becomes a VLDL remnant, IDL. And also the lipids exchange exists between the HDL and IDL and LDL, right? So the rest of the triglyceride that in the IDL or LDL that being transferred to HDL, and the HDL basically is the cholesterol-rich lipoprotein, means that HDL has a lot of cholesterol esters, right, in its core. So cholesterol esters can be transferred from HDL to IDL. So eventually IDL lose the triglyceride but gain, obtain a lot of cholesterol esters from HDL. So then eventually become the LDL, right? So LDL, you can see almost no triglyceride, but had a lot of cholesterol esters. So LDL is a cholesterol-rich lipoproteins. So finally, the LDL can be picked up by the liver and also peripheral tissues. So the cholesterol can be, transported to the liver and peripheral tissue for use. But if we have too much cholesterol being transported to the peripheral tissue, particularly for artery wall and macrophages, that will cause some problem, right? Atherosclerosis will happen. So we will have some specific session talk about the disease condition when we have something wrong with the lipoprotein metabolism, right? Okay, so LPL. We actually talk a lot about LPL, right? So you guys already know the main the important function of LPL is to hydrolyze triglyceride in both chylomicron and the VLDL, right? And which tissues or in which tissues LPL is synthesized? Which tissues can synthesize LPL? Adipose and muscle, right? So two major tissues. LPL can be synthesized in two major tissues, adipose tissue and muscle. And LPL is basically secreted, right? Secreted outside cells. It's not stay inside the cells. So into the wall of capillary endothelium within the tissue. I mean, it's still in the tissue, not 100% going to the circulation, but it's in the the wall of capillary endothelium in all the adipose tissue, muscle, or heart muscle, right? The LPL is not present in the liver. So liver has its own lipase. Remember we talked about, right? Hepatic lipase. So have a similar function, but the location is different. And the function of LPL is to hydrolyze the triglyceride and release the fatty acids that can be transported into the tissue, right? And the target of the LPL is the triglyceride within chylomicron and VLDL, right? And the purpose is, we mentioned earlier that triglyceride cannot be directly taken up by the tissue because they're too large, right, the molecule. So it need to be break down to fatty acids. So fatty acids are able to be taken up by the cells and tissue. So this is the reason why we need lipoprotein lipase to break down triglyceride, right, to help triglyceride get into the cells. And LPL in muscle or in the adipose tissue actually have a different activities, right? I mentioned earlier, because this enzyme in two different tissues had the low affinity for triglyceride in muscle and high affinity for triglyceride in adipose tissue. So so that's the reason why, you know, when high the fat conditions, the adipose tissue LPL is increased, and the fasting condition, muscle LPL activities increase, right? So during fasting, LPL activity is higher in which tissues? Muscle, right? Muscle. And in fat conditions, what tissues have high activity of LPL? Adipose tissue, right? So try to remember that, you know, the differences between adipose tissue, muscle in terms of LPL activities and function. And LPL activity requires the presence of a ApoC2, right, on the both VLDL and the chylomicron. So insulin basically can stimulate LPL synthesis and secretion from which tissues? Insulin increase, right, in the fat condition. So which tissues LPLs can be stimulated by insulin? Adipose tissue, yes, adipose tissue. So here we have two questions, and I think it should be easier for you, right? Now, so how are triglycerides in VLDL and chylomicron metabolized or mobilized? Yeah. Which enzyme is important, is needed for mobilize triglyceride in VLDL chylomicron? LPL, right? It's LPL. Huh? And which enzyme is the key? I mean, the same question similarly, you know, for LDL and chylomicron triglyceride metabolism. It's LPL, right? LPL is critical. Okay, more information about the LPL in adipose tissue, which usually activated in the fat state, right? So as you can see, when we consume a diet, particularly dietary triglyceride, right, need to be transported to the adipose tissue for storage, and chylomicron is synthesized in the small intestine and packed triglyceride, which is initially from the diet, and to the chylomicron, right, and to the adipose tissue. So in the fat condition, adipose tissue LPL is increased, right? It's synthesized and released and can hydrolyze triglyceride in the chylomicron. So LPL is increased by insulin. So insulin is a very important hormone that can stimulate LPL synthesized and the secretion in the adipose tissue during the fat condition. right? And the fasting condition, the muscle, the triglyceride can be also transported by VLDL to the muscle. And muscle also has a LPL, but its activity actually upregulated or increased during the fasting condition. So, and LPL can also hydrolyze triglyceride, right? And release fatty acids and uptaken by muscle and for oxidation for ATP production during the fasting condition. So, and this fatty acids initially from, triglyceride initially from the lipolysis, right? From the lipolysis in the adipose tissue. So fasting condition, right, we look at catecholamine, like epinephrine levels increase, it can stimulate lipolysis or triglyceride breakdown to fatty acids, and this fatty acids can be picked up from the circulation by the liver and packed resynthesized triglyceride and packed to VLDL, right? So VLDL and transport the triglyceride to the muscle and break down fatty acid again and pick up by the muscle and utilize in the muscle during the fasting condition. Right? Any questions? And I also wanted to mention that actually you raised that question. I think it's very important. I just wanted to clarify. I mean, during the fasting, you remember the liver also makes ketone bodies, right? So the fatty acids that picks up by the liver, that from the lipolysis, right, in the adipose tissue. So fatty acids can have several destination. Like first, fatty acids can be oxidized by breakdown to acetyl-CoA through the beta oxidation, right? And acetyl-CoA can be utilized for ketone body synthesis during the fasting condition. And some part of the fatty acids can be repacked to the triglyceride and sent it to the muscle and other tissue, right, for directly utilized as an energy source, right? So multiple, the metabolic pathway that occurred during the fasting. Any questions? Is that clear? Yeah. Mm-hmm.
What is it that stimulates?
Oh, what it stimulates? The catecholamine. Catecholamine can stimulate the LPL synthesis secretion in the muscle, right? So, and insulin basically stimulates synthesis of secretion of LPLs in adipose tissue, right? So they, regulates by different hormones in different tissue. Okay, so good cholesterol and bad cholesterol. I think we always thinking the HDL is a good cholesterol, right? And LDL is or VLDL, chylomicron, the kind of bad cholesterol because they have higher levels of triglyceride and cholesterol. So HDL is considered a good cholesterol because HDL can transport cholesterol from peripheral tissue back to the liver for disposal. So this is the main function of HDL. So that's why HDL is considered good, right? We wanted to remove extra cholesterol from in the body and to dispose it. So liver is a place that, the cholesterol can be disposed, either secreted through the bile acid, right? And non-HDL, like bad cholesterol, LDL and VLDL, and they actually, particularly LDL, is considered bad cholesterol because LDL actually deposit cholesterol into the peripheral tissue, particularly artery wall, right? So that is the bad thing. Now let's talk about the cholesterol-rich lipoproteins, LDL and IDL. So LDL, by its name called cholesterol-rich lipoprotein, that means that LDL core lipids basically contains more cholesterol esters, right? So the main lipids in the core of LDL core are cholesterol esters, but also have some triglyceride, right? And apoproteins on the surface of LDL is ApoB100. Pretty simple, right? Only has a one apoprotein, ApoB100, on the surface of LDL. And IDL is basically VLDL remnant. And the core lipids also include cholesterol ester, triglyceride, and apoprotein has two apoprotein, ApoB100 and also ApoE, that are on the surface of IDL. So in terms of the function or metabolism, LDL, the main purpose of LDL is to take up to deposit the the cholesterol into the peripheral tissue. So LDL can be taken up by the peripheral cells through the so-called LDL receptor mediated mechanism, right? So LDL has to be recognized by LDL receptor on the peripheral cell surface, then be able to being taken up, right, into the cell. So LDL receptor, again, it's very important, the key that control the LDL uptake and also control the LDL levels in the circulation, which makes sense, right? If LDL receptor for some reason have a decrease, then cell cannot take up enough LDL into the cell. So then more LDL will remain in the circulation. So I raise the circulating LDL levels, right? And also I mentioned that two important apoproteins, ApoB100, ApoE, are important for recognition of LDL receptor. It's also very important for controlling the rate of LDL uptake into the cells, right? So LDL receptor expressed mainly in the liver, but also other peripheral tissue as well. And I just mentioned the number of LDL receptor in the liver that determines the uptake of LDL, right? So it is the number of a receptor, LDL receptor, that control LDL concentration or levels in the circulation, right? And this actually involved the two uptake process involved two lipoprotein, right? ApoB100, ApoE. Does that make sense? Right? Any questions? Okay, now let's talk about LDL receptor. So LDL receptor is basically a single pass transmembrane protein that localize, that this just represents a cell surface membrane, right? So this is the LDL receptor. So LDL receptor basically is localized on the surface of the cell membrane. And LDL receptor has the ApoB100 ApoE binding site, and this for example this the represent the LDL lipoprotein, right? So LDL lipoprotein has the ApoB100, right, on its surface. So the ApoB100 on the LDL surface that can be recognized by LDL receptors, so that allowed the LDL to bind LDL receptors, and once LDL binds this receptor, then the whole membrane, cell membrane, can be internalized, right? So the whole receptor together with the LDL be internalized into the cells. So this is the process that how cell take up LDL, right? That requires LDL receptors and ApoB100 ApoE proteins on the LDL, right? More the process of LDL uptake and just a different version, right? You know, the cell membrane and LDL receptors, and LDL and binds LDL receptors and can be internalized. And LDL receptor actually can recycle, right? Can recycle, you know, membrane back to the cell surface and for the next turn of the LDL uptake. And LDL, which is lipids, right? And also together with apoprotein on the cell surface that can be eventually hydrolyzed to release cholesterol, free cholesterol, and protein can be broken down to the amino acids inside of cells. And cholesterol, once it's released in the free form from the LDL, that can be either re-acidified back to the cholesterol ester and stored in the inside of cells, right? And for use either for for refresh the membrane structures, right, or the other functions. So we need a cholesterol, but again, we don't need too much cholesterol, right? So, so acidification of cholesterol and the formation of a cholesterol ester, I just mentioned, right? The cholesterol, free cholesterol, cholesterol ester, they kind of balanced in the cell. So inside of cell, the two form free cholesterol, cholesterol ester are present in the cells, and they need to be balanced. So free cholesterol need to be reacidified to cholesterol ester. There is enzyme inside of cell called ACAT, the acyl-CoA cholesterol acyltransferase, ACAT, right? So this enzyme is catalyze the formation of cholesterol ester from free cholesterol and free fatty acyl-CoA. So cholesterol esters can be used for synthesis of bile acid and steroid hormones, right, in the liver, and also can be packed into the core lipids, right, in the different type of lipoproteins. And if the cholesterol can be, can be if cholesterol is too much in the cells, particularly in the macrophages, then cholesterol can be can be used to synthesize called atheroma, right? You know, cholesterol can be accumulate in the macrophages and becoming the firm cells, right? And firm cells is the basis of the atheroma, for cholesterol, for atherosclerosis, right? All right, so HDL. And HDL is another type of the cholesterol-rich lipoproteins, right? So core lipids in HDL is also cholesterol esters, right? Cholesterol ester, and more cholesterol ester and less triglyceride. And apoproteins on the HDL are the ApoA1 and ApoA2. And HDL is also synthesized in liver and also intestine. So what is the function of the HDL? HDL is considered good cholesterol, right? So what is the function? Why HDL is good? Cycles, right? Yes, the so-called reverse transport cholesterol pathway. So because HDL can transport cholesterol from peripheral tissue back to the liver, so then can keep a peripheral tissue cholesterol level low, right? And to remove the extra cholesterol from the body and to deposit in the liver. So liver can metabolize cholesterol and remove the extra cholesterol out of the body, right? So this is the reason. I mean, it's a good cholesterol. So the pathway for the cholesterol function is called reverse cholesterol transport, right? So reverse cholesterol transport is transport cholesterol from peripheral tissue back to the liver, and this reverse transport process is accomplished by HDL cycle, right? So HDL cycle means that HDL actually has two different forms. One is called alpha HDL, and the other form is the pre-beta HDL. And these two different type of HDL has a different lipid composition, right? And these two type of HDL can be interconverted, right? Let me see. So so I just give you kind of example here. So alpha HDL is called a full dump truck, right? I mean, HDL you can consider is just a truck that can pick up some unnecessary cholesterol from the peripheral tissue, right, and transport it to the liver. So alpha HDL is called a full dump truck. That means that they have almost a full load of the cholesterol, right? So if you can see the size, it's much bigger than compared to the beta HDL, right? So beta HDL basically is empty, truck. That means almost no lipid core, no lipids, right? No cholesterol, but only phospholipids, the surface lipids, and also apoproteins. And the alpha HDL, you have a full loaded of cholesterol, right? So it has in addition to the surface phospholipids and apoproteins, and also have you know, the core lipids, basically as the cholesterol, right? And alpha HDL basically give the cholesterol after it initially picks up from the peripheral tissue, right, to the liver. And beta HDL, because it's like empty trap, right, so basically it picks up cholesterol from peripheral tissue and forms alpha HDL, right? Yes.
What does FC stand for?
FC,
FC is
FC is the free cholesterol, and PL is phospholipids, right? So receive the free cholesterol and the phospholipids both, right, from the peripheral tissue and become the alpha HDL. So basically alpha HDL and beta HDL is the same, you know, the lipoprotein budget in the different stage, all right? So this, I think a diagram gives you a better idea how the reverse cholesterol transport happen, right? So the peripheral tissue, for example, artery wall, and you know the macrophages and the liver, right? So all this represents something happening in the plasma, in the circulation. So pre-beta HDL is just a surface, right? Phospholipid surface and no lipid core as considered empty tract. So they can pick up the free cholesterol and the phospholipids from peripheral tissue, from like, you know, macrophage on artery wall. And then once it's received that lipids from peripheral tissue and it's become a alpha HDL, right? You have a, you know, size is bigger because it contains the lipids, right? And then it becomes larger during the, you know, the transportation in the plasma. So, and this is a conversion is the process is also involved in multiple actually reactions. There are multiple enzymes, factors, protein that also involved in the you know, the conversion of alpha HDL from beta HDL. So eventually alpha HDL gets all the lipids from the peripheral tissue and arrive the liver, right? So the cholesterol can be taken up by the liver, and so all the cholesterol ester being picked up by the liver. So eventually, the alpha HDL then become back to the pre-HDL, you know, and empty tract again, and then go back to the peripheral tissue and pick up more, you know, cholesterol, lipids, and then, you know, send it to the liver. So this kind of going the cycles. So once you finish one cycle, then you will see the lot of cholesterol can be removed from the peripheral tissue, right? Okay, so these are the important proteins, factors, enzymes that involved in this reverse cholesterol transport process, or conversion between, you know, all the HDL cycles. We call it the same thing. One is ABCA1. I guess you guys don't have something that I wrote in red, right? So ABCA1 is ATP binding cassette transporter A1, right? This is a transporter, and this transporter is located on the cell membrane of peripheral tissues, like macrophages cell membrane or artery wall cell membrane, right? ABCA1 is a transporter, right? Lipid transporter. So what is this ABCA1 does is to remove the cellular cholesterol from the peripheral tissue and to load it free cholesterol onto the pre-beta HDL, right? That makes sense. So they function as a cholesterol efflux pump. So, transport free cholesterol from peripheral tissue to the beta HDL. And the second protein is called SRB1 or scavenger receptor B1. And this receptor is located on the liver cells, and the function of this receptor is to facilitate the uptake of cholesterol esters from, from HDL, right, in the liver. And the third one is called LCAT. So LCAT is the lectin cholesterol acyltransferase, and this protein LCAT is present in the plasma, right? So the function of LCAT is to convert free cholesterol into cholesterol ester in the plasma, in the alpha HDL in the plasma, right? So remember the beta HDL basically pick up the free cholesterol, right, from the peripheral tissue, and this free cholesterol needs to be reacidified to cholesterol ester that can be internalized to the core. Otherwise, the free cholesterol always present on the surface, right? So in order to be internalized to the lipid core in the HDL, free cholesterol that initially pick up from peripheral tissue need to be acidified to cholesterol esters. So this acidification process occurs in the plasma in the alpha HDL that requires this enzyme LCAT, right? Okay, so and another protein is called a CEPT, cholesterol transfer protein, and CEPT is also present in the plasma. And remember, we talk about the lipid exchange between HDL and LDL, VLDL, right? And this protein, CEPT protein, is a protein that facilitates this exchange, lipid exchange between HDL and LDL, IDL, VLDL, right? Okay, so the last protein is called ACAT. I think we'll talk about this like a couple of slides early. ACAT is also the enzyme that catalyzes the acidification of free cholesterol to cholesterol esters, but the location is different from the LCAT. The ACAT basically is localized inside of cells, right, to catalyze acidification of a free cholesterol to cholesterol ester inside of cells. So try to remember all this type, different factors, enzymes, what are their functions and how they are involved in the HDL cycles or reverse cholesterol transport process, right? Yes. Alpha HDL, in the alpha HDL. Beta HDL basically had nothing, right? No cholesterol. So once it's picked up, and then it becomes small alpha HDL, and then the larger alpha HDL.
And the exchange is also between the alpha and the beta, the HDL, right? the exchange of cholesterol and triglycerides.
Yeah. Triglyceride and cholesterol ester exchange between, yeah, right. Okay, so I guess let's stop here today.